Hi, let's talk about a chapter called Principle of Inheritance and Variation of Class 12. And firstly, I have to introduce myself. I'm a NEET student. I'm reappearing in NEET. I had last three attempts, and this is my last attempt I will give. And so I was thinking to share my reading skills and how to read the chapter quickly and to understand this faster we can. So I'm making this video, hope it will help you all. I'm following two books. One is the prominent NEET NCERT book, and the another one is PW's Dr. Vipin Sharma's this book. The mind maps given in that, or mind maps, yeah, short notes are very helpful to read quickly. So I'm starting with the notes here given. Let's start, and let's finish the chapter with lots of enthusiasm in 30 minutes. Termination we should know to read the whole chapter. Firstly, gene or factors. Next, alleles, homozygous, heterozygous, phenotype, genotype, Punnett square. So, what are these things? We know what is gene, and there's nothing to say about it. We can just tell it they are the units of inheritance by which the parents' characters are inherited in their offsprings. And alleles are, if we take a character like color of human skin, and there can or we can take the color of flower. So there can be many colors, uh, but the character is the color of flower. So one character, but it can have many contrasting trait or alleles. uh, it may be violet, red, white. So these contrasting characters are called traits or alleles, and the character is called itself character. Homozygous means it's pure. Heterozygous means dissimilar pair of alleles. Phenotype, external and morphological appearance of character. Genotype, genetic makeup of an organism. And Punnett square is that square I'm showing you. And this is called the Punnett square, by which we can make up the how many offsprings and how are their types. We can measure analytically. And let's come to Mendel's law of inheritance. So you should be aware that Gregor Johann Mendel, for whom we can now understand the principle of inheritance and variation, he took garden pea or Pisum sativum plants, and his experiments was for long 7 years from 1856 and to 1863. It should be remembered very clearly, and proposed law of inheritance. And he took 14 true breeding peas plants varieties. So 14 true breeding peas plants varieties he took as pairs which were similar except for one character with contrasting trait. It means in one time he took only one concentrated character. So as I say that in monohybrid cross, while taking any character, he is taking only the height, either it is tall or dwarf. So it is what is for one character with contrasting trait. It's mean just that monohybrid cross we see here. And let's read the characters it took. Stem height, flower color, flower position, pot shape, pot color, shape, seed color. And the contrasting trait, tall. Here the left hand sides are dominant traits and the right hand sides are recessive traits. For stem height, he took tall and dwarf. Flower color are in peas are violet and white. Flower position are axial and terminal. Pod shape inflated and constricted. Pod color green and yellow. Seed shape round and wrinkled, and seed colors are yellow and green. It should be remembered you don't need to confuse in the seed color and the where it is pot color. But in generally we consider the green pot color and green shades as the natural, as the good one. But it is for our economical purpose, for our need, for our taste, but the dominant one seed color is yellow. वो तो हमारी नीड है, क्योंकि हमें ज़्यादा ग्रीन ग्रीन चीज़ें ही खाना पसंद है, इसीलिए हम वो ग्रीन सीड्स और ग्रीन पॉट कलर वाला पीज ही उगाते हैं. And now let's come to reasons for Mendel's success. Mendel, firstly, I told that he took one character at a time. He concentrated only one character at a time. Secondly, his experiments had a large sampling size. This gave greater credibility to the data that he collected. He used statistical analysis and mathematical logic for analyzing the results. वो यूं ही कोई कैरेक्टर ये होगा वो नहीं लिए थे, वो प्रॉपर मैथमेटिकल लॉजिक एनालिसिस किए थे, जिसलिए वो रिजल्ट्स उतने परफेक्शन से आया उनके पास. The characters selected by Mendel were present on different chromosomes. ये इसका मतलब है वो जो कैरेक्टर्स लिए थे एक क्रोमोसोम पे दोनों कैरेक्टर वाला नहीं लिए थे. जितने कैरेक्टर्स लिए थे वो सभी कैरेक्टर्स अलग अलग क्रोमोसोम पे अलाइन करते थे. His references are confirmed from experiments on successive generations of his test plants. Now, inheritance of one gene. I need to break up some things. You can think that why I am taking this book only and not taking the NCERT. Wait, I will come to NCERT also, but here are the some things that are hidden in NCERT. We have to find very time taking it is in NCERT, so I'm covering it up with this book, and trust me, this book is so helpful. Now come to study of inheritance of one character at a time in an organism is called as monohybrid cross. So which I talked about previously, that in monohybrid cross we are taking only one character, means tall, we are taking here height, either it is of tallness or dwarfness. So this is called monohybrid cross. And phenotype means, told you previously, that the morphological character that can be seen from the outside, and genotype, the genotypic character that is inherited in the offspring. So the phenotypic ratio we get here, it is 3:1, and genotypic ratio is 1:2:1. Now, law of dominance. Trust me, this is so simple that once you understand it, you don't need to revise it after and after. You will just remind it very finely. Here, the story of tallness, dwarfness means the story of traits and alleles begin, and it will continue through the whole chapter. Now, we will talk about law of dominance, law of segregation, nextly incomplete dominance, codominance, law of independent assortment, chromosomal theory of inheritance, but one by one. You should have patience a lot, because we have to continue this in just 30 minutes. Now take monohybrid cross. In monohybrid cross, we are taking the character is height. Height of what? Height of pea plant. And the traits we taking, traits or alleles, we taking tall, pure, dwarf, pure. Tall, pure, dwarf, pure, capital T, small t. Let's make gamut of these. Small t, small t, small t, small t. From here, we can also understand the law of segregation. Law of segregation, what? It is just when the gametes are formed, the one trait is contributed in the gamete formation. जैसे कि यहाँ पर टी था, होमोजाइगस कंडीशन था, होमोजाइगस कंडीशन में दोनों ही टी टी है। सो एक गैमेट पे आया टी, एक गैमेट पे आया टी। जो कैरेक्टर होगा उनका ट्रेट्स इंडिविजुअली सेग्रीगेट करके गैमेट पर जाएंगे। वो लिंक्ड नहीं होगा। लिंक्ड मतलब वो ओवरलैप नहीं करेगा, वो इंडिपेंडेंटली जाएगा अगर इंडिपेंडेंट जीन हो तो। अगर इसके जगह होमोजाइगस ना होकर हेटेरोजाइगस होता तभी इक्वली डिस्ट्रीब्यूशन ऑफ ट्रेट्स होता। अगर वो ये होता सपोज कैपिटल टी एंड स्मॉल टी, देन गैमेट फॉर्मेशन के समय इक्वली डिस्ट्रीब्यूट होता ये ट्रेट्स। एक जगह जाता कैपिटल टी, एक जगह जाता स्मॉल टी। Let's come back to this. In F1 generation, F1 generation है what we say, तो क्या आया, टी 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 टी. Both phenotypic and genotypic ratios are same, and this is we can't make ratio because if we ratio them both are 1 1 1 1 and वो सब एक ही कैरेक्टर शो करेगा। यहाँ पर अब हम बात करेंगे अबाउट लॉ ऑफ डोमिनेंस। लॉ ऑफ डोमिनेंस की वजह से जो यहाँ पर मिक्सअप हुआ, यहाँ पर होमोजाइगस था, यहाँ पर आके ये हेटेरोजाइगस कंडीशन हो गया। तो यहाँ पे ये कोई ब्लेंडिंग नहीं शो कर रहा है, पर क्योंकि कैपिटल टी और स्मॉल टी एक साथ है ये हेटेरोजाइगस कंडीशन पे है। पर हेटेरोजाइगस कंडीशन पे होने के बाद भी ये कोई मिनिमम मतलब मीडियम हाइट नहीं शो कर रहा है। लाइक कोई पी प्लांट बहुत ज़्यादा लंबा है, कोई पी प्लांट बहुत छोटा है, ड्वार्फ है। तो ये सब जो F1 प्रोजेनीज जो होंगे वो सब उतने टॉल और उतने ड्वार्फ के मिडिल का कोई हाइट नहीं लेगा। आइदर वो टॉल होगा, आइदर वो ड्वार्फ होगा। But as the capital T is here, which is the dominant trait, so all pea plants will be of tall character. So all those 4 plants, all those progenies will have the larger height. Now make a Punnett square. F1 progeny. Now let's make its gametes. एक में आएगा कैपिटल टी, एक में आएगा स्मॉल टी, जो मैंने पहले अभी बात किया था ना लॉ ऑफ सेग्रीगेशन के बारे में। तो यहाँ पर वो ब्लेंडिंग नहीं शो कर रहा है। ये जो फिर से गैमेट बनाएगा तब इक्वली गैमेट में ये ट्रेट्स डिस्ट्रीब्यूट हो जाएंगे। यहाँ पर भी आएंगे कैपिटल टी स्मॉल टी। आई नो माई हैंड राइटिंग इज़ नॉट सो गुड बट नॉट सो बैड ऑल्सो, you can manage it by yourself. So this Punnett square, we will just do this type of thing and write it down. Capital T, capital T, capital T, small t, capital T, small t, small t, small t. It is pure, it is pure, pure, pure. It is heterozygous, it is heterozygous. सो पहले मैंने बताया था हेटेरोजाइगस पे क्या शो करेगा, लॉ ऑफ डोमिनेंस की वजह से जो डोमिनेंट ट्रेट होगा वो शो करेगा, और जो होमोजाइगस होंगे उस पे जिसका जो होगा वो शो करेगा। यहाँ पर टॉल का ट्रेट है तो इट वुड बी टॉल, यहाँ पर ड्वार्फ दोनों ही है सो इट विल बी ड्वार्फ। एंड द रेशियोज वी गेट, फेनोटाइपिक रेशियो विल बी, ये तीनों टॉल्स हैं। So phenotype and genotype 1:2:1। जेनोटाइप ये तीनों आलादा जेनेटिक मेकअप है, अलग अलग। So we are done with the monohybrid cross. Now from Ncert we will read law of dominance and law of segregation. Yes, law of dominance in the page 75, and I'm using the old NCERT book. Law of dominance. Characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates over the another one. So one is dominant and another one is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. What it means that वो जो डोमिनेंस लॉ ऑफ डोमिनेंस जो है उनका जो डोमिनेंट ट्रेट है वो F1 प्रोजेनी में शो कर रहे हैं. सभी सभी प्रोजेनीज पे वो डोमिनेंट जो ट्रेट है उसका नेचर शो हो रहा है बाहर से फेनोटाइपिक कैरेक्टर्स. एंड जो F2 प्रोजेनीज है वहाँ पर भी जो हेट्रोजाइगस है वो भी डोमिनेंट जो ट्रेट है वो शो कर रहा है और जो प्योर डोमिनेंट वाला जीन था वो भी डोमिनेंट ट्रेट शो कर रहा है. It also explains the proportions of 3:1 obtained at the F2 as I have done here. Here it is, and the genotype is, phenotype is 3:1. Threes are tall and one is dwarf. Then this is law of dominance. Now law of segregation. So from here it is अच्छा ये भी पढ़ लेते हैं. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered. Do not show any blending when in the F1 progeny. जहाँ पर वो हेटेरोजाइगस कंडीशंस शो कर रहा है होमोजाइगस पेरेंट्स से हेटेरोजाइगस प्रोजेनी हो रहा है वहाँ पर कोई ब्लेंडिंग नहीं हो रहा. And that both the characters are recovered and that's why both the characters are recovered in F2 generation. वो जो F2 generation पे हम पेरेंटल जो कैरेक्टर था प्योर टॉल प्योर ड्वार्फ वो वाला भी वापस आ रहा है और जो डोमिनेंट शो कर रहा है उसके वजह से वो जो हेट्रोजाइगस है कैपिटल टी स्मॉल टी वो भी शो कर रहा है. So from this that point you can use as the law of segregation. Now, incomplete dominance. So remember two lines. One is the 3:1 रेशियो पे जो कंट्रीब्यूट कर रहा है वो है लॉ ऑफ डोमिनेंस एंड लॉ ऑफ सेग्रीगेशन बोथ. तो अगर ऐसा क्वेश्चन आए कि 3:1 वो जो F2 जनरेशन पे रेशियो मिल रहा है उस पे किसका डोमिनेंस है वो माने किसका ज़्यादा कंट्रीब्यूशन है कौन सा लॉ की. तो जैसा कि NCERT पे लॉ ऑफ डोमिनेंस की जगह ये लाइन दिया हुआ है तो फर्स्ट चॉइस विल बी law of dominance and if there is more than one option to choose then we can use both the laws. नहीं तो law of dominance से ही काम चला लेना पड़ेगा. Now incomplete dominance. Incomplete dominance is something that the plant is neither showing the dominant trait nor showing the recessive trait but in between of those traits. As I told you that pea plants can be large, can be dwarf, but cannot be in between of these heights, cannot be of medium height. But incomplete dominance is something telling about that the height can be in between of those tallness and dwarfness. But it is the scientist is Carl Correns in 1903 done experiments on Mirabilis jalapa. ये दो नाम है वो याद रखना पड़ेगा. Mirabilis jalapa, डॉग फ्लावर एंड स्नैपड्रैगन. स्नैपड्रैगन की साइंटिफिक जो नेम है वो है एंटीरहिनियम मेजस. Antirrhinium majus. So the scientist is Carl Correns. So till now we have known about two scientists, Gregor Johann Mendel and Carl Correns. When a cross between two purebred, yeah, I told you that स्नैपड्रैगन का एक रेड फ्लावर लिया और एक वाइट वाला फ्लावर का प्लांट लिया, दोनों प्योर है। जब F1 जनरेशन है उस पे हम जानते हैं कि हेट्रोजाइगस जीन्स पाए जाते हैं। वहाँ पर वो जो heterozygous flowers there would be present, all those flowers will be of pink, neither red neither white, but in between of these two colors, and it is of pink, all will be pink, F1 progeny. And in F2 the two parental flowers are recovered, so here no blending occurred। F1 पे सभी करेक्टर्स, सभी जो सभी जो स्नैपड्रैगन्स थे वो सब फ्लावर की कलर पिंक पाए थे ड्यू टू लॉ ऑफ सेग्रीगेशन, क्योंकि वो जो एलील्स थे वो इंडिपेंडेंटली सेग्रीगेट किए थे। दैट्स व्हाई वी आर गेटिंग द पेरेंटल कैरेक्टर्स बैक इन द F2 generation। Here phenotypic and genotypic ratio both will be 1:1, 1:1। You can remember it just as Tay-Sachs disease is an example of the result of incomplete dominance, and Tay-Sachs disease is an autosomal recessive disorder। Laterally we will read about it। Now, codominance. Incomplete dominance is something that दोनों वो जो कलर है पेरेंटल कलर वो दोनों ही नहीं शो कर रहा है। इन द मिडल ऑफ दोज कलर्स कोई एक कैरेक्टर शो हो रहा है, बट कोडोमिनेंस इज समथिंग दैट बोथ द कैरेक्टर्स विल एक्सप्रेस देमसेल्व्स इन द प्रोजेनी। कोडोमिनेंस पे जो हम कैरेक्टर लेते हैं वो है ABO ब्लड ग्रुपिंग इन ह्यूमन। ओ का कोई सुगर नहीं पाए जाते हैं सरफेस पे, बट बोथ A and B have their own surface antigens present on RBC surface, which are A antigen and B antigen। Due to these, those are both these are A and B are dominant, and O is null because O do not contribute in producing any antigen over the surface of RBC, and the blood grouping is controlled by gene I। For A we get IA, for B we get IB, and for O we get small I, and it is we can consider it as null। So as O is not contributing in producing any antigen, that's why both IA and IB are completely dominant over small I or O blood group। So in the blood group of AB, both A and B contribute in expressing the character। O doesn't show any character, and A itself shows its character when present itself only। B shows its character, and when A and B both present in one blood group, then they both show their character, neither is suppressed। So the big difference, big difference you should remember, you should not forget about that incomplete dominance is something that neither of the parental characters is expressed, and codominance is something that both the parental characters are expressed। Now back cross और reciprocal cross के बारे में हम बात करेंगे। बैक क्रॉस जो है वो जो F1 प्रोजेनी पे जो पाए जाते हैं, वी गेट द प्रोजेनीस इन F1 जनरेशन, उनका क्रॉसिंग किए जाते हैं आइदर ऑफ द पेरेंट्स। अगर वो होमोजाइगस डोमिनेंट पेरेंट हो तो हम उसको आउट क्रॉस बोलेंगे, अगर वो होमोजाइगस रिसेसिव पेरेंट हो तो हम उसको टेस्ट क्रॉस बोलेंगे। सो बैक क्रॉस के in back cross we get two things. एक है out cross and another one is test cross. In out cross we cross F1 progeny with homozygous dominant. In test cross we cross with F1 progeny with homozygous recessive parents, and both the phenotypic and genotypic ratio we get is 1:1. And what is reciprocal cross? Reciprocal cross means एक बार फीमेल से किए जाएंगे, एक बार मेल से किए जाएंगे. अ पेयर ऑफ क्रॉसेस इन विच मेल एंड फीमेल सेक्सेस आर रिवर्स्ड. मतलब अगर टॉल मेल लिए थे, वो नेक्स्ट जो हम रेसिप्रोकल क्रॉस करेंगे उस पर टॉल फीमेल लिए जाएंगे और ड्वार्फ मेल लिए जाएंगे. नाउ inheritance of two genes, which is called dihybrid cross. In dihybrid cross we take 4 contrasting traits and 2 characters. So 2 characters and 4 contrasting traits are round, wrinkled, yellow, green. Character is seed shape and seed color. So यहाँ पर ये जो गैमेट्स, यहाँ पर गैमेट्स इंडिपेंडेंटली असॉर्ट कर रहे हैं, सेग्रीगेट कर रहे हैं. तो एक आर से एक आर आया, एक वाई से एक वाई, स्मॉल आर से स्मॉल आर, स्मॉल वाई से स्मॉल वाई. गैमेट पे आप सोच सकते हो कि स्मॉल आर, कैपिटल आर, कैपिटल वाई ऐसा क्यों नहीं हुआ? क्योंकि ये डाईहाइब्रिड क्रॉस हम लिए हैं और इस पे राउंड येलो 2 कैरेक्टर्स हैं जो हम लेंगे, और वो कैरेक्टर्स का जो जीन है वो हमको तो एक साथ ही लेना पड़ेगा ना. तो वैसे ही हम डाईहाइब्रिड क्रॉस कर सकते हैं, क्योंकि गैमेट्स तो हमें एक ही बनाना है. इससे गैमेट्स 2 गैमेट्स बनेंगे, 1 RY 1 RY, इस पे RY, इस पे RY. सो F1 जनरेशन पे जो आया राउंड येलो, एंड ये इतना बड़ा चार्ट यू डोंट हैव टू रिमेंबर, जस्ट मल्टीप्लाई द प्रीवियस वन जो मैंने बताया था. फेनोटाइपिक रेशियो जो मोनोहाइब्रिड क्रॉस पे था 3:1, उसका अगर आप स्क्वायर करेंगे, मतलब 3:1 इंटू 3:1, देन यू गेट 9:3:3:1. एंड जो जीनोटाइपिक है वो था मोनोहाइब्रिड पे 1:1. अगर 1 को दो अगर 1:1 को 2 बार आप मल्टीप्लाई करोगे तो ये पाओगे 1:1:2:4:2:1:2:1. So that's why you don't need to remember it. It's just a short trick I recalled from Bipin sir's lecture, whom this book is, Dr. Bipin Kumar Sharma, wonderful teacher. Now come to study, and just you need to remember the more phenotypic ratio will be of round yellow, then round green, wrinkled yellow, wrinkled green. The recessive one will get the least number and the dominant one will get the more number, and in between the recessive dominant will rely. Now law of independent assortment. Again we will do it from the Ncert book. Yeah, law of independent assortment, you can study it from here, पेज नंबर 80, और यहाँ पर छोटे करके दिए हुए हैं, ये भी हम क्यूट सा देख सकते हैं. When 2 characters are combined in a hybrid, segregation of one pair of traits is independent of the other pairs of traits. Combined in a hybrid, segregation of one pair of traits is independent of the other pair of traits. The above dihybrid cross between homozygous dominant smooth yellow seeded with homozygous recessive plants proved this law. वो जो हम ये लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट कहीं पे मिलता जुलता है लॉ ऑफ सेग्रीगेशन के साथ में. क्योंकि ये ब्लेंडिंग नहीं कर रहा है, ये इंडिपेंडेंट ही है, अपना ट्रेट इस गैमेट पे कंट्रीब्यूट किया है। राउंड वाला एक ट्रेट एक गैमेट पे, येलो वाला एक ट्रेट एक गैमेट पे कंट्रीब्यूट किया है। ये राउंड और येलो एक दूसरे के साथ ब्लेंडिंग नहीं कर रहा है, पर ये प्रेजेंट है अपना ट्रेट लेके एक गैमेट के पास। नाउ don't need to study. Yeah, chromosomal theory of inheritance. Chromosomal theory of inheritance, if we study it from NCERT, it will of great benefit. We can study the story here. मेंडल जो अपनी वर्क पब्लिश किए थे उनके टाइम से वो बहुत आगे चल रहे थे, दैट्स वाई उनके काम को कोई रिकॉग्नाइज नहीं कर पाए थे, remain unrecognized till 1900. Firstly, communication was not easy and couldn't widely published, so scientists उतने ध्यान नहीं दे पाए थे उनके काम पर 1900 पे तीनों साइंटिस्ट डेव रीस, कॉरिन्स and von Tischermark independently rediscovered Mendel's results on the inheritance of characters. Also, by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. यहाँ पर मार्क करोगे, ये तीनों साइंटिस्ट के वजह से ही मेंडल को रिकॉग्निशन मिले थे, एंड दिस वर कॉल्ड क्रोमोसोम्स जो डिस्कवर हुए थे। 1902 पे Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. So, chromosomal theory of inheritance was given by Sutton and Boveri in the year of 1902. In the year of 1900, Mendel's study was rediscovered due to Davries, Corrins and von Tissermark. ये दोनों लाइंस इम्पोर्टेंस है, ये और ये वाला। और वन थिंग है यहाँ पर, ये जो पेज 83 है वहाँ पर, Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal segregations with Mendelian principles. Sutton and Boveri ने मिल के क्रोमोसोमल थ्योरी ऑफ इनहेरिटेंस दिए थे, पर कौन सा दोनों चीज़ से, मेंडेलियन प्रिंसिपल्स एंड क्रोमोसोमल सेग्रीगेशन से क्रोमोसोमल थ्योरी ऑफ इनहेरिटेंस हमको मिला था। एंड अनदर साइंटिस्ट हियर केम T.H. Morgan, Thomas Hunt Morgan. Thomas Hunt Morgan did experiments with the Drosophila melanogaster, the fruit flies. Experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, and why he took only fruit flies? Because they complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Now let's recall the scientists and the year of their discoveries with me. Firstly, 1856 से लेकर 1863 तक ग्रेगर जोहन मेंडल, his experiments with the pea plants. Next, Carl Coren's 1903 on Mirabilis jalapa, experiment of incomplete dominance. Then comes 1900 पे Dave Rees, Correns and von Tissermark, by whom we could rediscover the work of Mendel. And then in 1902, the Sutton and Boveri chromosomal theory of inheritance with the Mendelian principles and chromosomal segregations. Now, linkage and recombination. You can confuse in linkage and recombination, but once you listen it calmly, you will understand as soon as possible. Linkage and recombination. Till now, we have read about the law of segregation, law of independent assortment, whether we could see that the characters didn't blended with each other. But now the thing come that they will blend. Blend means a thing recombination and linkage will come here. Let's start. Morgan hybridized yellow-bodied, white-eyed, which is a dominant character, to brown-bodied red-eyed males, we call it wild type, and intercrossed their F1 progeny. He observed that the two genes didn't segregate independently of each other, and the F2 ratio deviated very significantly from the 9331 ratio due to linkage and recombination expected when the two genes are independent. वो रेशियो कब आएगा जब दोनों जीन्स इंडिपेंडेंट होंगे. बट जब हम पढ़े थे पहले कि अगर जीन्स इंडिपेंडेंट होगा तभी लॉ ऑफ सेग्रीगेशन वर्क करेगा वहाँ पर. बट अगर जीन्स इंडिपेंडेंट नहीं होगा, जीन्स एक दूसरे के ऊपर लिंक्ड है, then it will not show independent assortment and deviate from this 9331 ratio. What is this linkage and recombination is? Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. Morgan attributed this due to physical association of linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Non-parental gene combinations जो होंगे उसको ही हम नाम देंगे recombination. So mark it here. जो नॉन पेरेंटल जीन कॉम्बिनेशंस होंगे उसको हम नाम देंगे रिकॉम्बिनेशन. एंड रिकॉम्बिनेशन कब कब होंगे? जब when some genes were very tightly linked, they will show very low recombination. When the genes are very loosely linked, they will show higher recombination. And we will also notice that the parental gene types will be more than the non-parental types always, and the non-parental gene combination comes due to the termination recombination. As if it is tightly linked as white-eyed and yellow-bodied, they are very tightly linked to each other in the chromosome, so they will show only 1.3% recombination, while white-eyed and miniature wings, they are not tightly linked. They are located on a far distance in the chromosome. That's why they will show a very more recombination of 37.2%. His student Alfred Strativert used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. So, who mapped their position on the chromosome in the linkage and recombination? Alfred Strativert was the student of whom? Student of T.H. Morgan. नाउ NCERT पे ये जो 84 नंबर पेज है इस पे बहुत सारे चार्ट्स दिए हुए हैं. यहाँ पर क्रॉस ए पे येलो बॉडीड एंड वाइट आइड विच आर प्रेजेंट ऑन पास. They are tightly packed and they are on दूर, so they are loosely packed, so they will show more recombination type of 37.2% and they will show less recombination type only 1.3%. Now, polygenic inheritance and pleiotropy. Polygenic inheritance. Inheritance of characters in which one character is controlled by three or more genes. जैसे कि ह्यूमन स्किन and human height. Human skin means human skin color, and human height is controlled by three or more genes. Intensity of character depends number of dominant allele. Dominant alleles are contributory in nature. Now pleiotropy. In this, a single gene can exhibit multiple phenotypic expression. In polygenic inheritance, it was multiple genes carrying out uh, one character, and in pleiotropy, one gene contributing over multiple phenotypic expression. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. जैसे कि starch synthesis in pea seed. Due to starch synthesis in pea plant, in pea seeds, the size and shape varies. If starch synthesis is done more, then it will have a round shape and a large size, and if starch synthesis is in less amount, then it will get a wrinkled shape and small size. Now we will study about sex determination. Determination was given by Henking in 1891. So another scientist is added whose name is Henking, and 1891 sex determination he did on few insects. Before doing sex determination, we need to study these, that in Drosophila and human the genotype of female is XX, and genotype of male is XY. In birds, it is in female ZW and in male is ZZ. In insects, it is XX and genotype of male XO. O means one gene is missing from here. It is null. So, male is haploid in insects. Male is or haploid. It is a wrong termination. We should rather use term that one gene is absent in male in insects and the female is homozygous. In Drosophila and human, female is homozygous, but in birds, the female is heterozygous. Whether in honeybee, the female is diploid but the male is haploid. Now comes the interesting part, sex determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosome an individual receives. honeybee का जो मेल्स हैं वो अनफर्टिलाइज्ड एग्स ऑफ फीमेल से फॉर्म करते हैं. That's why they don't have a father but have grandfather and grandsons. Whether the females are formed, the queens are formed by the mating of uh, female female egg and male sperm. That's why the females are diploid and the males are haploid. And it is the most interesting part. The male gets 16 chromosome and female get 32 chromosome. I was wondered when I was reading this, but it is so wonderful. Now the part another part interesting, mutation. And mutation के बाद जो डिसऑर्डर्स रहे हैं वो ही सिर्फ बाकी है. उसके बाद हमारे ये चैप्टर we will done with it. Let's study mutation and then we will do some talks about next chapter and we will be done. Mutation can be chromosomal mutation or can be gene mutation. What is mutation? Sudden inheritable change in an organism is called mutation. Chromosomal mutation कैसा है? It is due to structural and numerical changes in chromosomal structure. Chromosome structural changes of chromosome involve following types: deletion or duplication. Deletion. A part of chromosome is lost from terminal part or from intercalary part. What is duplication? It occurs due to the addition of a part of chromosome. So chromosomal mutation is either a deletion of one part of chromosome, either adding up of one part of chromosome is duplication. And gene mutation can be deletion, insertion, frame shift mutation, or point mutation. Deletion, delete of one nucleotide, one or more nucleotide base pairs. Insertion, addition of one or more nucleotide base pairs. Frame shift mutation, both deletion and insertion can lead to frame shift mutation. Now it is interesting. Frame shift mutation containing both deletion and insertion. One nucleotide is deleting and another is adding up. That's why the total base sequence shifts laterally either in the forward direction or in the backward direction. Next, point mutation. Mutation arises due to the change in a single base pair of DNA. Due to the change in a single mutation of a base pair, and that's called point mutation. जैसे कि सिकल सेल एनीमिया ग्लूटामिक एसिड सब्सटीट्यूट कर देते हैं वैलिन को, दैट्स वाई सिकल सेल एनीमिया इज फॉर्म्ड. यहाँ पर सम पेडिग्री एनालिसिस है. ये मेल है, ये फीमेल. सेक्स अनस्पेसिफाइड, अफेक्टेड इंडिविजुअल, मेटिंग इन बिटवीन रिलेटिव्स. ये 5 ऑफस्प्रिंग्स है. इनका जो मेल बेबी है वो अफेक्टेड है. इनका फीमेल और मेल दोनों बेबीज हैं. ऑटोसोमल, अब कुछ डिसऑर्डर्स के नाम पढ़ लेते हैं जल्दी से. ऑटोसोमल डोमिनेंट ट्रेट. ऑटोसोमल डोमिनेंट ट्रेट में है मायोटोनिक डिस्ट्रॉफी. ये ऑटोसोमल डोमिनेंट. ऑटोसोमल रिसेसिव, टाई सेक्स डिजीज एंड सिकल सेल एनीमिया. एक्स लिंक्ड ट्रेट डोमिनेंट. ये क्रिस क्रॉस इनहेरिटेंस शो करते हैं. मेल से एक जनरेशन पे मेल पे होगा तो दूसरे जनरेशन पे फीमेल में होगा, फिर तीसरे जनरेशन पे ये मेल पे हो जाएगा. ये है एक्स लिंक्ड डोमिनेंट. एक्स लिंक्ड रिसेसिव में है हीमोफीलिया ए और क्लासिक हीमोफीलिया. एक्स लिंक्ड रिसेसिव. सो ऑटोसोमल डोमिनेंट में है मायोटोनिक डिस्ट्रॉफी. ऑटोसोमल रिसेसिव टेसेक्स सिंड्रोम एंड टेसेक्स डिजीज एंड सिकल सेल एनीमिया. एक्स लिंक्ड रिसेसिव में है हीमोफीलिया ए और वाई लिंक्ड जो ट्रेट है वो सिर्फ मेल पे ही अपीयर होगा. नाउ मेंडेलियन डिसऑर्डर्स. मेंडेलियन डिसऑर्डर्स वो सभी डिसऑर्डर्स है जिनको हम पेडिग्री एनालिसिस में डाल सकते हैं. Now let's talk about the recessive disorders and autosomal recessive and the sex-linked recessive disorders. And firstly, you should know the mostly genetic disorders are recessive. So one is sex-linked recessive and autosomal recessive. In sex-linked recessive, only two disorders are there. One is hemophilia and another one is color blindness. And in autosomal recessive, one is sickle cell anemia, 2 is thalassemia, and 3 is phenylketonuria. Let's understand these diseases. सिकल सेल एनीमिया, ग्लूटामिक एसिड सब्स्टिट्यूट कर देते हैं वैलिन, सब्स्टिट्यूशन ऑफ ग्लूटामिक एसिड बाय वैलिन। सब्स्टिट्यूशन पहले मैंने थोड़ा गलत बता दिया था, तो माफ़ कर देना, कि वैलिन glutamic acid is substituted by valine at the sixth position of beta globin chain, at the sixth position of beta globin chain. हीमोफीलिया तो वो क्लॉटिंग फैक्टर से रिलेटेड है, क्लॉटिंग फैक्टर ठीक ठाक काम नहीं करते हैं, दैट्स वाई ब्लीडिंग नहीं बंद होते। नाउ फिनाइल कीटोनुरिया, the affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. phenylalanine into tyrosine जो कन्वर्ट करते हैं वो जीन अफेक्टेड है, वो एंजाइम अफेक्टेड होते हैं। कलर ब्लाइंडनेस is X-linked recessive. That's why 8% males and only about 0.4% females are. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. पहले हमने पढ़ा था कि about 0.4%, so many less number of females are attacked by this disease than the males ones. Red-green color blindness is seen in color blindness of X-linked recessive. Now thalassemia, either due to mutation or deletion in alpha or beta chain. क्रोमोसोम 16 पे जो HbA1 and 2 है उसके लिए अल्फा थैलासेमिया होते हैं और बीटा थैलासेमिया होते हैं क्रोमोसोम 11 पे HbB जिनके वजह से। And the major difference between sickle cell anemia and thalassemia is that thalassemia is a quantitative problem but sickle cell anemia is a qualitative problem. Now chromosomal disorders में एन्यूप्लॉइडी और पॉलीप्लॉइडी पाए जाते हैं। एन्यूप्लॉइडी वो है क्रोमोसोम्स ड्यूरिंग सेल डिविजन सेल साइकिल रिजल्ट्स इन द गेन और लॉस ऑफ क्रोमोसोम्स called aneuploidy. During cell division, gain or loss of chromosome. But what is polyploidy? Failure of cytokinase. After telophase stage of cell division results in an increase in a whole set of chromosome. So during cell division cell cycle, loss or gain of chromosome, or during cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosome, that's called the polyploidy. Now Down syndrome, Klinefelter syndrome and Turner syndrome. And trisomy, monosomy, additional copy of a chromosome may be included in an individual. 2n+1 हुआ तो trisomy, 2n-1 हुआ तो monosomy. अगर एक जीन एडअप हुआ वो ट्राइसोमी हो जाएगा, अगर एक जीन ओमिट हुआ तो वो मोनोसोमी हो जाएगा। व्हाट इज़ डाउन सिंड्रोम? 21st क्रोमोसोम का 3rd कॉपी आ जाए तो एक तो 1 है, ये 21 क्रोमोसोम दोनों होगा, एक मदर से आएगा एक फादर से आएगा, बट अगर तीसरा कोई कॉपी आ जाए तो उसको हम डाउन सिंड्रोम बोलेंगे। ये लैंगडॉन ने डिस्कवर किया था, लैंगडॉन डाउन। इस यहाँ पर मेंटल रिटार्डेशन, पार्शियली ओपन माउथ, पाम क्रीज पाम हो जाएगा, ब्रॉड एंड स्मॉल फेस हो जाएगा। नेक्स्ट क्लाइनफेल्टर सिंड्रोम, यहाँ पर मेल इंडिविजुअल पे एक ज़्यादा एक्स क्रोमोसोम एडअप हो जाएगा, 44 + XXY, दैट्स वाई मास्क्यूलाइन डेवलपमेंट होगा। गायनेकोमैशिया मीन्स डेवलपमेंट ऑफ ब्रेस्ट, फेमिनाइन कैरेक्टर भी डेवलप हो जाएगा। एंड इंडिविजुअल जो मेल इंडिविजुअल होगा वो स्टेराइल होगा और मेंटली रिटार्डेड होगा। नेक्स्ट टर्नर सिंड्रोम, यहाँ पर एक एडअप हो रहे हैं, यहाँ पर एक ओमिट हो रहे हैं। 44 + X, only one chromosome and another one is removed from the female one. So it is Klinefelter syndrome is for male, Turner syndrome is for female, and Down syndrome क्रोमोसोम नंबर 21 पे ट्राइसोमी ऑफ 21 इट कैन ऑकर इन बोथ मेल एंड फीमेल। तो टर्नर सिंड्रोम पे क्या होगा, ड्वार्फ होगा, मेंटली रिटार्डेड होगा, फीमेल्स का ओवरीज जो होगा वो रूडिमेंट्री हो जाएगा एंड लैक ऑफ अदर सेकेंडरी सेक्सुअल कैरेक्टर्स। सो वी आर डन विद द चैप्टर very finely. I would be waiting for your response to this video, and please tell me if I do more videos on the biology chapters and which chapter is seem very difficult to you. I will simplify it to you in just 30 minutes, and let me know in the comment section how the lecture was and what chapter I should take in my next video. Thank you. Tata.